Hello friends, welcome to our YouTube channel. In this video we have brought to you mock test number 7 for Food Corporation of India based on Biotechnology syllabus. So we'll try to come up with more test series for FCI as per syllabus related to agriculture. So stay connected with us and subscribe to our YouTube channel for upcoming mock tests. Now let's start today's mock test. Question number 1. The ribosome of bacteria is, the options are 30S, 80S, 70S or 20S. The correct answer is option C, 70S. So the ribosome of bacteria is 70S. 70S ribosomes are present in the bacterial cell which has 30S smaller subunit and 50S larger subunit. Question number 2. The recombination in which the genetic material transfer through virus is called as dash. The options are transduction, transfusion, transformation or conjugation. The correct answer is option A, transduction. So the recombination in which the genetic material transfer is through virus is called as transduction. Basically, in transformation, a bacterium takes up a piece of DNA floating in its environment. In transduction, DNA is accidentally moved from one bacterium to another by a virus, and in conjugation, DNA is transferred between bacteria through a tube between cells. Question number 3. A plant virus with double-stranded DNA is, the options are Gemini virus, Colimovirus, nanovirus or both A and B. The correct answer is option B, Colimovirus. So Colimovirus is a plant virus with double-stranded DNA. Example, cauliflower mosaic virus. Question number 4. Penicillin was discovered by, the options are Anthony van Leeuwenhoek, Alexander Fleming, Robert Koch or Louis Pasteur. The correct answer is option B, Alexander Fleming. So penicillin was discovered by Alexander Fleming. Alexander Fleming was a Scottish physician scientist who was recognized for discovering penicillin in the year 1928. Question number 5. The cell wall of bacteria is made up of, the options are cellulose, glucose, peptidoglycan or peptide. The correct answer is option C, peptidoglycan. So the cell wall of bacteria is made up of peptidoglycan. The cell walls of gram-positive bacteria are composed predominantly of peptidoglycan. In fact, peptidoglycan can represent up to 90% of the cell wall. Question number 6. The life cycle of bacteriophage result in death of the bacterial cell is called as dash cycle. The options are lytic, lysogenic, apoptosis or induction. The correct answer is option A, lytic. So the life cycle of bacteriophage result in death of the bacterial cell is called as lytic cycle. In lytic cycle, the bacteriophage infects a bacterium, hijacks the bacterium to make lots of phages and then kills the cell by making it explode. Question number 7. Yeast is dash. The options are multicellular bacteria, unicellular bacteria, multicellular fungus or unicellular fungus. The correct answer is option D, unicellular fungus. So yeast is unicellular fungus. Yeasts are unicellular fungi reproducing asexually by budding or fission and sexually by spore formation. Question number 8. Which type of RNA molecules make up more than 80% of the total RNA in the cell? The options are rRNA, mRNA, tRNA or none of the above. The correct answer is option A, rRNA. So rRNA is the type of RNA molecule which makes up more than 80% of the total RNA in the cell. Ribosomal RNA are found in the ribosomes and account for 80% of the total RNA present in the cell. Question number 9. The HOH bond angle in water is dash. The options are 103 degree, 104.5, 106 degree or 107.5. The correct answer is option B, 104.5 degrees. So the HOH bond angle in water is 104.5 degree. Question number 10. The chromosome complement of 2n-2 is dash. The options are monosomy, trisomy, nullisomy or double nullisomy. The correct answer is option C, nullisomy. So the chromosome complement of 2n-2 is nullisomy. In nullisomy, 2 chromosomes are missing and the chromosomal composition is represented by 2n-2. Question number 11. Which of the following enzyme was first isolated and purified in the form of crystals? The options are urease, pepsin, insulin or amylase. The correct answer is option A, urease. So urease is the enzyme which was first isolated and purified in the form of crystals. Question number 12. Which of the following is the bioindicator of air pollution? The options are lichens, E. coli, fungi or algae. The correct answer is option A, lichens. So lichens is the bioindicator of air pollution. Let's know more about it. Lichen, which consists of a symbiotic relationship between a fungus and an algae, is sensitive to atmospheric pollution, including nitrogen and sulfur emissions that lead to acid rain, as well as toxic lead and mercury emissions. This sensitivity makes lichen a valuable biological indicator of air quality. Question number 13. In bacteria, sexual reproduction occurs through which mechanism? The options are conjugation, transformation, transduction or all of these. The correct answer is option D, all of these. So in bacteria, the sexual reproduction occurs through all the three, which is conjugation, transformation and transduction. So in a bacterial cell, sexual reproduction occurs via 3 different methods: conjugation, transformation and transduction. Question number 14. Plasmid that carry genes responsible for bacterial conjugation is dash. The options are F plasmid, R plasmid, Col plasmid or L plasmid. The correct answer is option A, F plasmid. So plasmid that carry genes responsible for bacterial conjugation is F plasmid. Question number 15. In India, food safety standards are decided by dash. The options are FPO, FSSAI, Ministry of Health and Family Welfare or FCI. The correct answer is option B, FSSAI. So in India, food safety standards are decided by FSSAI. Food Safety and Standard Authority of India is a statutory body established under the Ministry of Health and Family Welfare, Government of India. The FSSAI has its headquarters at New Delhi. Question number 16. DNA synthesis takes place at dash stage of interphase. The options are S, G1, G2 or H. The correct answer is option A, S. So DNA synthesis takes place at S stage of interphase. The S phase of a cell cycle occurs during interphase before mitosis or meiosis and is responsible for synthesis or replication of DNA. Question number 17. Nucleoside is made up of dash. The options are organic base plus deoxyribose, organic base plus deoxyribose plus phosphate, deoxyribose plus phosphate or organic base plus phosphate. The correct answer is option A, organic base plus deoxyribose. So nucleoside is made up of organic base plus deoxyribose. A nucleoside consists simply of a nucleobase, also termed as nitrogenous base, and a 5-carbon sugar, ribose or deoxyribose. Question number 18. The first X-ray diffraction patterns of DNA were taken in 1938 by dash. The options are Rosalind Franklin, Watson, Crick or William Asbury. The correct answer is option D, William Asbury. So the first X-ray diffraction patterns of DNA were taken in 1938 by William Asbury. Question number 19. Chromosomes are smallest and thickest at dash stage. The options are metaphase, anaphase, prophase or telophase? The correct answer is option A, metaphase. So chromosomes are smallest and thickest at metaphase stage. So the best cell division stage to observe the shape, size and number of chromosomes is the metaphase. The chromosomes appear shortest and thickest during this phase and are arranged at the equator. Question number 20. The term biotechnology, bios plus technologia, is composed of dash words. The options are Latin and Greek, Latin and French, French and Greek, or two Greek words. The correct answer is option D, two Greek words. So the term biotechnologia is composed of two Greek words. Question number 21. The term biotechnology was coined by dash in 1919. The correct answer is option A, Carl Erke. So the term biotechnology was coined by Carl Erke in the year 1919. Question number 22. For the surface sterilization of explant, which of the following chemical is used? The options are sodium hypochlorite, mercury chloride, both A and B, or none of the above. The correct answer is option C, both A and B. So for the surface sterilization of explant, both sodium hypochlorite and mercury chloride can be used. Question number 23. MS medium, which is generally used as nutrient medium for plant tissue culture, was given by. The options are Gamborg, Herbertland, Carl Erke, or Murashige and Skoog. The correct answer is option D, Murashige and Skoog. So MS medium, which is generally used as a nutrient medium for plant tissue culture, was given by Murashige and Skoog. So MS medium was invented by two scientists named Toshio Murashige and Falk K. Skoog in the year 1962. While the two scientists were working on the discovery of plant growth regulators, it is the most commonly used medium in the tissue culture lab. Question number 24. Which of the following plant tissue culture technique is used to obtain haploid plants? The options are meristem culture, anther or pollen culture, ovule culture, or both B and C. The correct answer is option D, both B and C. So both anther or pollen culture and ovule culture are the plant tissue culture technique which is used to obtain haploid plants. Question number 25. Anther or pollen culture technique of plant tissue culture was given by. The options are Gamborg, Herbertland, Guha and Maheshwari, or Murashige and Skoog. The correct answer is option C, Guha and Maheshwari. So anther or pollen culture technique of plant tissue culture was given by Guha and Maheshwari. Question number 26. The first person to demonstrate the idea of totipotency of cell wall was dash. The options are Gamborg, Herbertland, Guha and Maheshwari, or Murashige and Skoog. The correct answer is option B, Herbertland. So the first person to demonstrate the idea of totipotency of cell was Herbertland. Question number 27. DNAs consisting of repetitions of extremely short units are called. The options are microsatellite, cDNA, codon, or anticodon. The correct answer is option A, microsatellite. So DNAs consisting of repetitions of extremely short units are called microsatellite. Question number 28. Dash is the technique which uses electric discharge to produce pores on cell membrane for intake of recombinant DNA. The options are electrophoresis, PCR technique, electroporation, or none of the above. The correct answer is option C, electroporation. So electroporation is the technique which uses electric discharge to produce pores on the cell membrane for intake of recombinant DNA. Question number 29. A DNA element which has the ability to move from one chromosomal position to another is called dash. The options are chromomere, jumping gene, transposon, or both B and C. The correct answer is option D, both B and C. So, a DNA element which has the ability to move from one chromosomal position to another is called jumping gene or transposon. Question number 30. By using recombinant DNA technology, dash gene is transferred to maize genotype, which is responsible for high amount of lysine in maize. The options are CK60, Norin 10, opaque 2 or cry 2. The correct answer is option C, opaque 2. So by using recombinant DNA technology, opaque 2 gene is transferred to maize genotype, which is responsible for higher amount of lysine in maize. Question number 31. Hybrids produced using nucleus of one parent cell and cytoplasm of both the cell are called dash. The options are vibrids, cybrids, top cross hybrid or none of the above. The correct answer is option B, cybrids. So hybrids produced using nucleus of one parent cell and cytoplasm of both the cells are called cybrids. Question number 32. Dash is the group of adjacent genes that are identical or related. The options are gene library, germplasm, gene cluster or none of the above. The correct answer is option C, gene cluster. So gene cluster is the group of adjacent genes that are identical or related. Question number 33. Which of the following is also known as reduction division? The options are mitosis, first meiotic division, second meiotic division or both B and C. The correct answer is option B, first meiotic division. So first meiotic division is also known as reduction division. Question number 34. Plasmid is independent dash autonomous molecule of DNA. The options are double-stranded, closed circular, self-replicating or all of these. The correct answer is option D, all of these. So plasmid is independent, double-stranded, closed circular, self-replicating autonomous molecule of DNA. Question number 35. The plasmid which replicates only when the chromosome replicates is dash. The options are stringent plasmids, relaxed plasmid, TI plasmid or all of these. The correct answer is option A, stringent plasmids. So the plasmids which replicates only when the chromosome replicates is the stringent plasmid, whereas relaxed plasmids replicate their own independently. Question number 36. Which of the following is tumor-inducing plasmid often responsible for crown gall induction in plants? The options are stringent plasmids, relaxed plasmids, TI plasmid or all of these. The correct answer is option C, TI plasmid. So TI plasmid is the tumor-inducing plasmid often responsible for crown gall induction in plants. Question number 37. Bacteriophage is a virus that lives in bacteria, first observed by Dash in the year 1940. The options are Ivanovsky, T.J. Burrell, Ruska or Stanley. The correct answer is option C, Ruska. So bacteriophage is a virus that lives in the bacteria, first observed by Ruska in the year 1940. Question number 38. In 1982, the first biotech plant, an antibiotic resistant dash was developed. The options are soybean, cotton, tomato or tobacco. The correct answer is option D, tobacco. So in 1982, the first biotech plant, an antibiotic resistant tobacco was developed. Moving on to the next question, dash technology could be used to prevent any gene flow between biotechnology and traditional crops in which plant produce sterile seeds. The options are BT technology, GMO technology, Terminator technology or none of the above. The correct answer is option C, Terminator technology. So Terminator technology is the technology which could be used to prevent any gene flow between biotechnology and traditional crops in which plant produce sterile seeds. Question number 40. Dash is an experimental application of biotechnology in which genetic engineering is used to create plants that can produce pharmaceutical proteins and chemicals. The correct answer is option B, biofarming. So biofarming is an experimental application of biotechnology in which genetic engineering is used to create plants that can produce pharmaceutical proteins and chemicals. Question number 41. The proportion of different genotypes in a random mating population is known as. The options are gene frequency, genotype frequency, genotypic variation or genetic recombination? The correct answer is option B, genotype frequency. So the proportion of different genotypes in a random mating population is known as genotype frequency. Question number 42. Random change in gene frequencies due to sampling error is called dash. The options are accidental mutation, genetic drift, genetic erosion, or genetic shift. The correct answer is option B, genetic drift. So random change in any direction in gene frequencies due to sampling error is called genetic drift. Question number 43. The unit of heredity material that controls expression of a particular phenotype is dash. The options are DNA, gene, chromosome, allele. The correct answer is option B, gene. So the unit of heredity material that controls expression of a particular phenotype is gene. Question number 44. The Human Genome Project was launched in the year dash. The options are 1980, 1973, 1990, or 1989. The correct answer is option C, 1990. So the Human Genome Project was launched in the year 1990. The Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up human DNA and of identifying and mapping all of the genes of the human genome from both the physical and functional standpoint. Question number 45. Which is a genetically modified crop? The options are BT cotton, BT brinjal, golden rice, or all of these. The correct answer is option D, all of these. So the genetically modified crops are BT cotton, BT brinjal, and golden rice. Question number 46. A double haploid plant is. The options are always vigorous, 100% homozygous, 100% heterozygous, or always true breeding in cross-pollinated species. The correct answer is option B, 100% homozygous. So a double haploid plant is 100% homozygous. Question number 47. In vegetative tissue and seeds, phosphorus is stored in the form of dash. The options are cysteine, phytane, trisodium phosphate, or calmodulin. The correct answer is option B, phytane. So in vegetative tissue and seeds, phosphorus is stored in the form of phytane. Question number 48. An enzyme involved in initiation of DNA synthesis during replication is dash. The options are telomerase, holoenzyme, RNA primase, or endonuclease. The correct answer is option C, RNA primase. So an enzyme involved in initiation of DNA synthesis during replication is RNA primase. Question number 49. The scientist who had given the name mitochondria, and the correct answer is option B, Carl Benda. So the scientist who had given the name mitochondria was Carl Benda. Question number 50. The country that deregulated golden rice recently to address the deficiency of vitamin A is dash. The options are India, Bangladesh, Philippines, or Indonesia. The correct answer is option C, Philippines. So the country that deregulated golden rice recently to address the deficiency of vitamin A is Philippines. Deregulation means a removal of government regulatory control from commodity. It means more freedom and more distribution of the commodity. That was all. Thank you friends for watching our video. Hope you find it useful. Please subscribe to our YouTube channel for more such videos. Thank you so much.